Now we are going to study the next topic, that is chemiosmotic hypothesis. Why do we study chemiosmotic hypothesis in photosynthesis? The reason is that we have to see how the ATP is generated. We had talked about phosphorylation, the cyclic and non-cyclic, both of them have been discussed, and we know that the outcome of that photochemical phase of photosynthesis was ATP and the reducing power, that is NADPH2. These two things were our outcomes from the photochemical phase. And one thing that we had studied, that when the electron moves through the electron transport chain, it comes down its gradient, it generates ATP. Now, before we start, I would like you all to move back in time and go to the understanding of topics of class 11, where we had studied how in electron transport chain of oxidative phosphorylation. Now the name is oxidative phosphorylation. That means the ATP generation would be driven by oxygen. We had studied in mitochondria, it was taking place while we studied respiration, that ATPs were generated by 5 different complexes which were present in the membrane of mitochondria, if you can recall it. If you cannot recall it, better do it, because that is the topic where we had studied how electron moves from one place to another, from one acceptor to another acceptor down the gradient, and in process the ATP is generated in the end because there is pumping of protons. So when we study chemiosmotic hypothesis, you have to remember that the scientist who gave it was called Michel, and remember this. Whenever you read it from somewhere, you're going to find his name. He was the one who proposed it. Chemiosmotic hypothesis, when we are studying in regard to photosynthesis, we are dealing with ATP generation from the photochemical phase, first thing. Okay, now he was the one who gave it. Second thing, as I told you, from your knowledge of previous class, you know that it is the proton gradient, the pumping of protons, which is responsible for jumping of electron from one acceptor to another acceptor, and of course that is driven by some or another redox reaction that is taking place between different complexes of the membrane. So we have to see the proton pump basically over here. Okay, you don't see these things actually happening inside the leaves, you cannot see them, but it is proposed that this actually happens from our knowledge of chemistry and from our knowledge of redox reactions and from our knowledge of what is located where inside the leaf. We have proposed a hypothesis. As the name suggests, it is chemi, that means something doing with chemistry, and it is dealing with osmotic concentrations as well. Other thing I told you that there is a proton pump. It would be similar to the one hypothesis where you studied how ATP was being generated in the oxidative phosphorylation while you studied respiration, but the only difference would be that the site would be different. What the site would be? In this case, because we are dealing with photosynthesis, because we are dealing with photochemical phase, the site would be chloroplast. Profuse knowledge of chloroplast you have by now, because you have studied in detail its structure. Now inside the chloroplast there is another site that we studied, that is thylakoid. Okay, now this chemiosmotic hypothesis that we study, the site of this particular process that is taking place, that would be involving proton pumps, and another thing, ATP generation, the site would be thylakoids. Okay, one thing that is to be kept in mind. Now because the ATP was generated in the cyclic photophosphorylation and the non-cyclic one, so that was photochemical phase. So certainly the two things that we have to study, and by any means we cannot skip them, is photosystems that we had talked about. So the PS2 and PS1 would also be there in the scene. Alright, so we have thylakoids, we have these two photosystems. Now, these two are to be studied, unlike that complex 1, complex 2, complex 3 and complex 5 that we had studied in oxidative phosphorylation. We are not going to study about them. We are going to talk about photosystem 2, we are going to talk about photosystem 1, we are going to talk about thylakoids, and because it is almost similar to what we have studied in respiration, what would be important in thylakoids would be the membrane of thylakoid. So, we have to deal with the membrane system of thylakoids, we have to deal with thylakoids of the chloroplast, we have to deal with photosystem 2 and photosystem 1. These are the things that we have to deal with. Now, one by one we are going to see that what actually happens in chemiosmotic hypothesis. ATP generation is required, photosystems are involved. Let's see what would be there inside the thylakoid where this chemiosmotic hypothesis has to be studied. Firstly, you have to consider thylakoid as a double membranous structure. Two membranes are surrounding a central lumen. Okay. This is the outer membrane, this is the inner membrane. The photosystems which have the pigment units or the photosynthetic units comprising PS2 and PS1 would be present over here. This is PS2, this is PS3, oh PS1, not PS3. PS2 and PS1 photosystems they are. This is lumen, this is stroma. Let's get rid of this name now. This is stroma of the chloroplast, okay, chloroplast that is present inside the plant cell. Now, inside the chloroplast there would be many thylakoids. One is PS2, one is PS1, inside there is lumen. Now, over here we are going to consider another unit that is F0 F1 particle, or you call it F0 F1 particle, something like this. Okay, that is present in the membrane, again the thylakoid membrane. This is what it looks like, here it is, and other name for it is ATP synthase. Okay, this particle. Another thing that is to be kept in mind, that inside the stroma, that is outside the membranes, there is an enzyme known as NADP reductase. It is present towards the outer membrane inside the stroma. Now, what we have to deal with over here is, as I told you, there has to be a proton pump that would be present. See, when the light falls on the photosystem, you have studied that the electron would be excited, it would move from its electron acceptor, the electron excited from photosystem 2 would go to cytochrome complex, that is cytochrome B, cytochrome 6, F, sorry, not 6, cytochrome B6F complex, it goes to PS1. This is what happens between two photosystems. The electron, it gets excited by photons which come over it, they are excited, there is the movement of electron from plastocyanin to plastoquinone. When this particular movement is taking place, when this photochemical process of photosynthesis is going on, that is non-cyclic photophosphorylation, we are not taking phosphorylation aspect, just we are taking the excitation of electron. This electron is going from the first acceptor to the third acceptor by cytochrome complex. This cytochrome complex, because of the energy changes which are taking place, it takes up protons from outside. Protons which are present inside the stroma. When it takes this proton, when the electron is accepted by cytochrome, the protons are also taken and that causes the energy changes. The proton goes to the next acceptor, that is plastocyanin, and these the electron goes to the next electron acceptor, and the protons which were absorbed by cytochrome, they are thrown inside the lumen. One thing. This is the first thing that you have to remember, because there is constant excitation of electrons from that reaction center of the photosystems, and those electrons are going to the photosystem 1 through the electron transport chain, where cytochrome lies as an acceptor. When this cytochrome takes up those electrons, in the process, side by side, it picks up the protons as well from the stroma in order to meet the energy requirements and attain stability. When the electron moves to the next acceptor, the protons which were accepted by cytochrome, they would be passed on inside to the lumen. Okay, this you have to remember, because when the cytochrome would take up the electrons, it would become negatively charged. In order to balance that negative charge, it would take up protons from stroma, not from inside but from stroma, okay, outside. So keep in mind that supposedly outside the electron number was 8, just for instance, 8 not electrons, 8 protons were there in the beginning. You can take it 80, you can take it 800, whatever your choice is. We are taking 8 protons. Now what happens is 2 protons have gone inside. So this is reduced to 6, sorry. Okay, and in the beginning inside the lumen there were in the same way 8 protons only. Okay, after this step what has happened? There we have got 6 protons, and over here by addition of 2, those 2 have come inside, it becomes 10 protons are present. Okay, there is a change, increase of protons inside the lumen. At the same time, as you know, that there is splitting of water in order to meet the charge requirements of photosystem 2, because the electrons are getting excited. Who provides electron to photosystem 2? The water molecules after splitting. It provides the electrons. So at the same time, there is splitting of water near the inner membrane, and what happens is that water breaks up into this process takes place, right? There is splitting of water that we had discussed. So there is increase of 2 more protons inside. So what is the number of protons now? It becomes 12. 12 protons are present inside, though it had started in the beginning, it was 8. Now this is the second step. So you have to remember splitting of water. I would write it as it is. It is H2O adds protons, electrons and nascent oxygen. This is the process that is taking place inside the lumen, as a result of which the number of protons increases inside the lumen. Then there is a third point. Come back to the electron that was excited from the photosystem as a result of the photochemical reaction that is taking place. Now this electron, when it reaches photosystem 1, it goes to agent X, it goes to reducing complexes of iron, and finally it reaches NADP reductase. When it comes in contact with NADP reductase, which is present on the outer membrane, it takes up protons, again protons from the stroma, and forms NADPH2 and forms the reducing power, which further leads to a greater decrease in number. So now compare these 3 points. First is that the electron got excited, it came in contact with cytochrome, which took the protons from stroma and threw them inside the lumen. Second is, inside the lumen, in order to meet the energy requirements of photosystem, which had lost its electrons, there is splitting of water and there is further addition of protons into the lumen. Third is that in the case of this photosystem process that is taking place, both photosystems, the electron that is excited from photosystem 1 goes to NADP positive in the presence of NADP reductase, which is present outside, not inside the lumen but outside on the outer membrane. That NADP reductase enables taking up protons from the stroma itself and fixing them into NADPH2. And in the beginning, as we had started with 8 protons in both lumen and the stroma, now at end, what do we have is over here the number has increased to 12 and there it has reduced to 4. So 3 steps are taking place, as a result of which there is a proton gradient that is being formed. Okay, you see a gradient, there are 4, there is 12, as much we started in the beginning with equal number, but now there is a gradient, and as a result of gradient, this F0 particle enables the movement of electron outwards, okay, because now they have the tendency to diffuse out. There would be diffusion of protons from F0 particle. When this proton which is moving out of F0, it comes in contact with F1 particle. Such is the nature of F1 particle that using the energy transitions which are taking place because of the proton outflow from here as a result of gradient, this F1 particle uses ADP and inorganic phosphate to form ATP. This is all, okay. We had discussed the 3 points which brought about the gradient. When the gradient is formed, the protons are bound to move out and form the proton pump. As a result of diffusion, this protons would come out, they would diffuse out, the pump would be formed, and F0 will enable that movement out. As soon as the proton comes in contact with F1 particle of ATPase molecule, the ATP synthase enables that energy transformation to be used by phosphate inorganic and ADP, that has 2 phosphate, that is adenosine diphosphate, combines with inorganic phosphate, and all the energy that this gradient was carrying along with it is fixed in the bond of phosphate that it forms with ADP. And in this way, ADP gets converted to a high energy ATP, and the energy that was brought about by the photon to this photosystem, that was taken up by the electron and then it moved from one place to another, gets stored over here. So this is what chemiosmotic hypothesis is all about.